Carrier-mediated diffusion is a passive transport that occurs because a carrier is facilitating that diffusion. So the cell isn't actually putting out the energy to make the work happen because a carrier protein is picking up the particle that needs to be moved and is actually moving it or is pushing it through a channel. So proteins are transported in the membrane, either in or out of the cell. Cell membrane does not allow these solutes to pass because they're too big or too polar. So if the cell membrane doesn't allow things to pass through it, a carrier protein can go grab it and pull it through, or open to allow it to go through. So it can make a bigger channel for these solutes to go through if the cell membrane does not allow it to go through. The transport proteins are specific for this particular solute. So specific proteins will allow passage or move specific things. Not all proteins move all solutes. And there are three types. There's facilitated diffusion, there's primary active transport, and secondary active transport. Those are all types of carrier-mediated diffusion. And the proteins can act as a channel to allow specific solutes to enter and exit the cell. So either they can open and close to allow one solute at a time to go through, they can grab and pull them through, or they can act as a channel where things can slide through. Carrier-mediated diffusion has specificity, and saturation can change the rate at which it happens. So in the carrier-mediated diffusion, this example, we have a ligand-gated channel. So the channel is going to be something that opens and allows things to pass through, just like a channel for boats. So in this case, we have these gated channels, and a solute or a ligand will come along, and if it's the right one to go through the channel, it will bind to the site on the channel, the receptor site, and cause the channel to open. When that channel opens, these solutes can move through. When the solutes are done moving through, the ligand will unbind and it will close the channel. Only specific ligands can open these channels. They are like keys. This key opens this channel. This key might not open the next channel. So it's very specific to each channel and each ligand. When the solutes travel through these channels, they are unchanged. Nothing happens to them. They just float through and they come out on the other side normal. They don't go through any chemical or physical changes. So when the saturation of a solute rises, they're going to transport faster because the concentration gradient is a lot higher. If we have the outside of the cell and we add a whole bunch more solute, it's going to want to move into the cell faster. Because remember, increasing the concentration of anything will increase the rate of diffusion. So this will happen until it gets to a transport maximum. The transport maximum is when all of the gates are full and the carriers are all busy moving solute. They can't move anymore. So it'll speed up until all the carriers are too occupied moving their solutes where they can't pick up extra solutes. That is the transport maximum. All the carriers are occupied and there's none running around loose to pick up any extra solutes. That will affect the rate of carrier-mediated diffusion. So there are three main kinds of carrier proteins. There are uniport, symport, and antiport. And their names kind of tell you what their function is. So a uniport protein carrier, or a uniport carrier protein, will carry one type of solute. That's why it's called a uniport. It has one port. Okay, an example of this is a calcium pump. It only carries calcium. A symport will carry two or more solutes at the same time in the same direction. So it's like carpooling. If one's going one way and another one needs to go the same way, a symport carrier can grab both of them and carry them through. Okay, so sym means together, like working together. So a symport, it works together to carry two. An antiport can carry two or more solutes in opposite directions. So anti means like against, right? So an antiport, it can carry two solutes in opposite directions. So one could be going into the cell and one could be going out of the cell, all at the same time. Okay, so an example of that is a sodium-potassium pump. When the sodium goes out, the potassium comes in. They go opposite directions. That's an antiport carrier protein. And if you look at this picture, the furthest left one, A, is a uniport. You can see only one thing is going through in one direction. B is symport. It's carrying two. It's carrying a substrate and this proton. They're going the same direction, but it is carrying two things. And then C is carrying two different things in different directions. So that would be antiport. So in facilitated diffusion, the carrier moves solutes down a concentration gradient. So it's just like diffusion, where the concentration is going to be higher, either inside or outside the cell, and the solutes are going to move from that area of higher concentration to the area of lower concentration. That is going to happen passively, so no ATP is going to be consumed by the cell. The solutes are going to attach to a binding site on the carrier. The carrier is going to change shape, open up, allow for passage of a certain size or certain shaped particle, and then it's going to enter it, and then the carrier is going to open up again or change shape so it can come out the bottom. So if you look at this picture, you can see that there is a bunch of these solutes in the extracellular fluid in the ECF. So the carrier goes into this, or the solute goes into this carrier. You can see the carrier has a slot big enough for one solute, or one particle. So it goes in. Once it gets into the site, it will bind at the site, which will activate the carrier to change shape or change conformation. It'll cause the carrier to close at the top and open at the bottom. Then the solute will move out into the cell, into the intracellular fluid. So this is how facilitated diffusion happens with carriers. And again, because it is diffusion, it's high concentration to low concentration, no energy because we're going down the gradient. Okay, so primary active transport occurs when cells move solute through a membrane against or up the concentration gradient. So we're going to be going from a low concentration to a high concentration, which is uphill, right? So it's going to take energy. The carrier is going to consume energy from the cell. And some examples of this are calcium pumps. They use ATP when expelling calcium from the cell where it's already more concentrated. So there might be less calcium inside the cell than outside the cell, but the cell needs to get that calcium out. So it's going to have to use energy to push it out because there's no concentration gradient that's going to favor moving it out. It's going to favor moving it in. And sodium-potassium pumps use energy when expelling sodium and importing potassium. So those are those antiport pumps that push one thing one way and the other thing the other way. And it's always going to be where one thing is going to be against a concentration gradient, if not both things. So it has to take energy, or it's got to consume energy to move things one of the two ways, if not both ways. So primary active transport, things are moving through the membrane against the concentration gradient, and they are consuming energy. So secondary active transport occurs when a carrier moves a solute through a membrane and indirectly consumes ATP. So an example of this is a sodium glucose transporter. It's a symport. So it's going to carry two things in at the same time, same direction. And if you look at the picture at the top, you can see there's a lot more glucose outside of the cell than inside. So the concentration gradient is going to work in the favor of moving the glucose into the cell. Okay. It might also work in the favor of moving the sodium into the cell because it looks like there's a lot more sodium in that area outside the cell than inside. So the symport carrier pulls them both in. They're going to travel down the cell, and then at the bottom, the cell is going to expel them back out. So the bottom, where the sodium-potassium pump is, it's pushing sodium out. But if you look at the bottom, there's a lot more sodium outside the cell in that area than inside. So the concentration gradient is the other way around. The sodium does not want to go out because it wants to stay in, because it's higher outside and it wants to come in to balance it, right? So the cell has to use energy to push that sodium out and brings that potassium in, because sodium-potassium pump, one goes one way, the other goes the other way. And it's going to expel energy while pushing out that sodium because that is going against the gradient or going uphill. So secondary active transport, it's not directly using the energy. It's happening indirectly. So in this example at the top, the glucose sodium is coming in without energy. That's a secondary active transport because that's not consuming the energy exactly. Because the sodium is coming in, the energy is being consumed. So it's not direct energy use while transporting molecules. So now we have vesicular transport. This is how things move large particles like fluid drops or a lot of molecules at one time through a membrane. They move them in bubble-like things called vesicles. So they have motor proteins that work as little motors, and they are ran by energy. So it definitely consumes ATP. It is active transport, and these things can either go in or out of the cell. So they can bubble around a group of items or a group of molecules, whatever they need, and carry it, kind of like luggage, either into the cell or out of the cell. They're ran by motor proteins that act as little motors, and they have to use energy. So by this time, we know that cells can both take in things and put out things. So let's talk about these processes. So the first process is endocytosis. And if we break this word down, endo means in or enter, cyte means cell, and osis just means condition of. So endocytosis breaks down to condition of entering or going into a cell. So endocytosis is when cells bring things in. And this can happen by phagocytosis, pinocytosis, or receptor-mediated endocytosis. So phagocytosis is when cells eat large solid particles. Okay, they can do these by using pseudopods, which are those finger-like projections that they can grow when they need them and then get rid of them. There are phagosomes, which are parts of the cell that are used to ingest things. And then macrophages, they also ingest large things. Cells can also take things in by pinocytosis, and this is called or referred to as cell drinking. So this is when cells take in liquid molecules. Membranes can cave around the liquid and kind of bubble around it and then break off into like a little vesicle. I remember phago and pinocytosis because Pinot sounds like Pinot, like wine. So it reminds me that it's drinking. So Pinot is wine, pinocytosis is drinking. The other one is eating, because it's not drinking, it's got to be eating. So pinocytosis is drinking and phagocytosis is eating of the cell. And then we have receptor-mediated endocytosis, which can be taking in anything when it's using receptors. So particles bind to specific receptor sites on the membrane, and then the membrane kind of bubbles around it and pulls it in. If you look at these pictures, you can see how the membrane kind of bubbles around and engulfs things like this. These are all parts of the membrane's job. They just are categorized by the type of thing that they're bringing in or if they're being helped. So solids is phagocytosis, liquids is pinocytosis, and if a receptor is binding to the membrane to like encourage it to take things in or activate the process of taking things in, it is receptor-mediated endocytosis. So here's another visual of receptor-mediated endocytosis. You can see that the receptors are binding to the membrane. And remember, receptors only bind to specific sites. Not just any receptor can bind to any site. They're very specified. So this receptor is made to bind to this site. So these receptors come in and they bind to the site. When they bind to the site, they activate the cell's membrane to kind of make like a bubble and start pulling those things in. Okay, it'll continue to pull them in until it kind of makes a vesicle, and then it will break off from the membrane and separate itself, and that bubble will go into the cell. And then the cell can consume it and do whatever it intended to do with it. This could be for energy. It could be because something needs to come in the cell so it can be modified. It could be a virus that the cell is trying to break down, a bacteria. It could be one of many things, but it will pull it into the cell to do whatever it has intended to do with it. So cells bring all these things in with endocytosis, but they also need to get things out. Okay, so cells have waste, or they have parts of them that are no longer functioning, or something has gotten into the cell that is causing damage to the cell, and they need to get it out. So the process of getting things out of a cell is exocytosis. Exo means to exit, cyte is cell, and osis is condition of. So when we break down exocytosis, we get the condition of exiting or leaving the cell. So exocytosis, in short, is just discharging material from the cell that is no longer wanted or needed. So transcytosis, and if you guys remember what trans means, it means like through or across or to the side. And cyte means cell, and osis is, again, condition of. So transcytosis is transport of material across a cell, okay, by capturing it on one side and releasing it on the other. So instead of going around the cell, it's going to go through the cell. Okay, so the receptor-mediated endocytosis is going to pull this thing in. The cell is going to then do exocytosis and push it back out the other side. Okay, so usually the things will go through unchanged. There's many reasons why it would go through. And it depends on the type of cell. It depends on what it's trying to accomplish. But in this picture, you can see we have blood, and it's going into the brain. So this is probably one of the most simple examples of transcytosis. This is going to be the blood-brain barrier, where in your brain, the brain fluid and the blood do not mix. There is a layer of tissue in between them that separates them. So anything that is in the blood that needs to get into the brain is going to have to go through this tissue. But again, we don't want blood in there. So whatever nutrients or whatever proteins, whatever needs to get to the brain's fluid is going to go through transcytosis. It's going to leave the blood, enter the tissue, and be pushed through by endo and exocytosis and out the other side. Just whatever needs to get through, the proteins, the nutrients, whatever it might be, not the actual blood. So transcytosis is transporting something through the cell, but it's not being eaten or taken in. It's literally just going in and coming right back out the other side. So the cytoskeleton of a cell is a network of protein filaments and cylinders. It gives the cell its shape. It works as a support structure. It can organize the contents. It can control movement and tell it where to go. It's basically the skeleton of the cell. So what your skeleton does for your body, this is going to do for the cell. It is made of microfilaments and microtubules. Okay, and cyto means cell. Skeleton means skeleton. So it literally breaks down to cell skeleton. So the cytoskeleton is made of microfilaments. There's a lot of microfilaments in our bodies, and we'll talk a lot about these when we get to the muscular system. But the microfilaments that make up the cytoskeleton are about 6 nanometers thick, and they're made of actin protein. They form a web. Then there's intermediate filaments, which are a little thicker. These are within skin cells, and they have protein and keratin in them. They will give cells their shape, and they resist stress. So they keep the cell nice in tone, and they resist stress, like pushing on it, squishing it, things like that. Microtubules are a lot thicker. They are 25 nanometers thick, and they consist of protofilaments made of protein tubulin. So these can radiate from the centrosome, and they can come and go. They maintain cell shape. They hold the organelles in place. So the parts of the cell that are in the cell are held in place by microtubules. Some things are like loose in the cytoplasm, but there are some things that are anchored down, and they're anchored down by microtubules. They act like railroad tracks for walking motor proteins, so they give a nice pathway for things to move in and out of the cell or around the cell. And they also make the axonomies of the cilia and the flagella. So if you remember when we talked about the cilia and the flagella, they were all made of rings, and they had microtubules. So that's what these things are. They are basically the framework for the cell skeleton and some of the cell structures, like the cilia, which are the hair-like things, and the flagella, which are the tails that are only on the sperm in the human body. And a microtubule, if you look at it, it kind of looks like a pull and peel Twizzler. All right, so a quick rundown on the structures of the cell. We're going to go over these a little more in depth in lab next week, but just a quick rundown of the function and why they are important for the cell to sustain life. So organelles are any internal structure of a cell that carry out specialized tasks. So inside of the cell, there are many organelles. There's the nucleus, the mitochondria, the endoplasmic reticulums, the Golgi. All these things are organelles. Okay, you have membranous organelles and non-membranous organelles. So membranous organelles are organelles that have their own plasma membrane. So they regulate what goes in and out of those organelles. And then there are non-membranous organelles. They don't have any membrane. So nothing is regulating what goes in and out of them. So probably the most important part of a cell is the cell nucleus. The cell nucleus is the control station of the cell. So the nucleus is usually the biggest organelle inside the cell. Most cells are uninuclear, meaning they have one. There are a few types of cells that have multi-nucleus, so they're multi-nucleated cells. And there are some cells that have no nucleus. Nucleoplasm is the material that is found inside the nucleus. So it is made of chromatin, and there are also nucleoli. So chromatin are DNA and protein composed threads, and the nucleoli are masses where ribosomes are produced. So here's a picture of the nucleus. It's in the middle. It is the control center of the cell. It holds all of the genetic material for the cell. Without the nucleus, the cell has no idea what its job is. So the nucleus is a membrane-bound or a membranous organelle. It has a membrane around it. It's really important to regulate what enters and exits the nucleus because the nucleus is the control center. If the nucleus goes down, the whole cell goes down. So making sure that bacteria and viruses do not get in the nucleus is important. But also, the DNA of the cell is stored in that nucleus. And we don't want that DNA just floating out loosely around in the cell and getting out of the nucleus. So the nucleus has a double membrane around it. So it has perforations in it by rings of proteins. And we'll talk about why there are perforations, why we need to be able to get to that DNA. It helps regulate traffic through the envelope, and it holds the two membranes together. But things do need to enter and exit, but it's very controlled what enters and exits this nucleus. So the nuclear envelope is supported by a nuclear lamina, which is a web of protein. It provides attachment points for chromatin, and it helps regulate cell life cycle. So chromatin play a very important role in mitosis, which is cell division, in how that DNA is carried on from one cell to the next. So when babies are made, when the egg is fertilized by the sperm, it has to split and duplicate and make more cells. That DNA has to be carried from one cell to the next, and chromatin play a huge role in that. And that's part of the cell life cycle. So this nuclear lamina is really important in regulating that and making sure it's occurring correctly. Right outside of the nucleus, we have the endoplasmic reticulum, or the ER. So the ER is a system of channels enclosed by a membrane. It is a membranous organelle, and it is all enclosed. The rough ER has ribosomes all over the outside of it. So there's a smooth and a rough ER. The rough one is like studded with ribosomes. So if you look at this picture, the darker pink, see how it has all these little bumps all around it? Those are ribosomes. Ribosomes are important because they make proteins. So the rough and the smooth ER are continuous with each other and the nuclear envelope. They are attached to the nuclear envelope, and then it flows into the rough ER and then the smooth ER. The rough ER produces phospholipids, which are important for the cell's membrane, and proteins. Protein synthesis can occur here, and they can be packaged and transported out of the cell from the rough ER. So the smooth ER has no ribosomes, which is why it's nice and smooth, but it is continuously attached to the rough ER. There is no ending of one and beginning of another. They flow together. In the smooth ER, steroids and lipids are usually synthesized here, and it can also detoxify alcohol and other drugs that are ingested. It can store calcium as well. Essentially, the rough and the smooth ER are different parts of a similar network. So they work in a similar way and they have a common goal, but they are different functioning parts. So now we have the ribosomes in the Golgi complex. So ribosomes are small chunks of protein with some RNA in them. They're found in the nucleoli, in the cytosol, and in the outer surfaces of the rough ER, as well as the nuclear envelope. The job of these is to read coded genetic materials from RNA and assemble amino acids into proteins that are specified by that code. So if you remember back, proteins fold very specifically to the job they need to do. The map for this folding comes from DNA. So the DNA has all the coding to fold these proteins the way they need to be folded so they work right. The only problem is the proteins do not know how to read DNA. The ribosomes that do the folding do not know how to read DNA. So RNA translates the DNA code into a code that the ribosomes and the protein can understand. So the RNA translates the message that is coded in the genes and tells the ribosomes how to fold the proteins. So when the ribosomes are folding the proteins, if they're folded correctly, it's conformation. Remember, when proteins are folded right, it's conformation. So once the proteins have been folded, they go to the Golgi. The Golgi's job is to put the finishing touches on the proteins and inspect them to make sure that they are folded correctly. So all new proteins from the rough ER go to the Golgi. The Golgi checks them, make sure that they look good. If they look good, they can be pushed out into the cell and then released wherever they need to go into the body. If they're not, they need to go back and be refolded. So now we have lysosomes. Lysosomes are packages of enzymes bound by a membrane. They are intracellular and they aid in digestion of proteins, nucleic acids, complex carbs, phospholipids, and other substances. Lysosomes can also perform autophagy, which means that it can digest parts of the cell that it is in that are not needed. And it can perform autolysis, which is cell suicide. It's when the cell purposely kills itself because there's too many of it or something is wrong with it. So lysosomes are like the little cleanup garbage men of the cell. They go through and they use endocytosis to engulf things that are not needed in the cell. They can eat bacteria, they can eat dead parts of things. Anything that is not necessarily needed or wanted, the lysosomes can get rid of. They can push things out of the cell as well. So lysosomes are like the cleanup of the cell. And the way I remember is because lysosome sounds like Lysol, and you use that to clean. They do perform the autophagy, like I said. If a cell has an extra organelle that it doesn't need or it's not working, the lysosome can digest it or eat it. And then it can do autolysis, which means cell death. But cell death. So it can kill the cell if it needs to. So peroxisomes are a lot like lysosomes, except for their function is to use oxygen to oxidize organic molecules. So when this happens, it produces hydrogen peroxide, which is why they're called peroxisomes. So they neutralize free radicals, detoxify alcohol, detoxify drugs, and they can get rid of a lot of bloodborne toxins. They can break down fatty acids. They can do all of these types of things, but in every process, they use oxygen and they produce peroxide. Okay, so peroxisomes break things down just like lysosomes, but they have a byproduct of peroxide, which is why they're called peroxisomes. These are really, really abundant in the liver and the kidney. And why do you think that is? What is so important about the liver and the kidney where they would need these type of cells? Well, the liver and the kidney function everything we take into our body. So a lot of medication, a lot of alcohol, bad foods, free radicals, all those things are filtered either by the liver or the kidney, depending on what it is. And there are a lot of these cells in both because they need to be there so they can break down these toxins that are taken in. If we didn't have the peroxisomes in this area, it would probably take a lot longer to break down the toxins if we could even break them down at all, which could lead to a lot more problems with toxins building up inside of our body. So when we have these proteins that become denatured, or we don't need them anymore, or something else is wrong with them, we got to get rid of them somehow. So these proteasomes are the protein destroyers when we don't need the protein anymore. So they're cylindrical organelles that get rid of extra proteins or proteins that have been tagged because they don't work or they're broken or something like that. So the proteins are tagged, and these things come around and they look for the tags. When they find the tags, they break down the protein that is tagged, and it's destroyed. It's destroyed and it's like disposed of. Get rid of it. It doesn't need to be in the cell if it's not working. So those are proteasomes, and they only break down surplus or unneeded proteins. So the mitochondria. This is where we make our energy. Okay, so cells have mitochondria in them, and this is how they make energy to do cellular functions. Okay, this is how energy is created, or this is where energy is created. They're usually the shape of a sphere or thread-like. They're surrounded by a double membrane. The inner membrane has folds in it. And in between those folds is the matrix. That's where the ATP synthesis happens, in the matrix. Inside the matrix, there are ribosomes, and there are enzymes that are used to speed up the ATP synthesis process. Multiple molecules of mitochondrial DNA. It's mtDNA is how you notate that it's mitochondrial DNA. And the mitochondria is usually referred to as the powerhouse of the cell because this is where the energy is created. This is where energy is extracted from organic molecules and transferred to ATP. So this is where sugar is broken down in the process of glycolysis. And then it's transferred into the matrix for the Krebs cycle and electron transport. All of these are processes of making energy, and they occur in the mitochondria, specifically in the matrix of the mitochondria. The mitochondria always looks like this with these little folds in it, so it's easy to identify because it has all the folds. And then the little purple dots inside are the ribosomes that are inside the mitochondria. So now we have centrioles. These are short, cylinder-shaped microtubules. So we talked about microtubules when we talked about the cytoskeleton, and they looked like those Twizzler pull-in peels, right? They look like plumbing poles that you'd see like on the back of a truck, right? Or PVC pipes. So centrioles are different because they were arranged in 9 groups of 3. So there's going to be 9 groups of 3 microtubules together, and they're going to be arranged in kind of a star pattern. These play a really important role in cell division, and they form the basal body of the cilia and the flagella, or like the core, the base, the inside body. And if you look at these pictures, you can see that it kind of looks like a star, and you can see the 3 microtubules in the 9 groups. So the last thing are cell inclusions. These are anything extra inside of a cell that is not an organelle, basically. So there's 2 kinds of inclusions. There are stored products or foreign bodies. So the cell can have something that it took in purposely and is storing. And this could be like glycogen granules, which is a form of sugar, pigments, or fat droplets. It might need these for something, or it may have just taken them in to store it for later. And then there are foreign bodies. So these are things that should not be in the cell that somehow got in. Things that usually do that are viruses or bacteria, debris from another cell bursting or being eaten or being destroyed. It can get those things inside the cell. Usually those things are not good to have in the cell. The cell usually would not purposely take them in. It would try to get rid of them, but sometimes they are stuck in the cell. They are never enclosed inside of a membrane, so they're always non-membranous, so they don't have their own membrane. And they're not essential for cell survival, so the cell does not need these things. In reality, foreign bodies are usually detrimental to the cell. So if a virus gets into a cell, it can hijack the cell and take over the cell and use the cell to make more viruses or to make the body sicker. So usually we don't want these foreign bodies inside of our cells because they're not really good. We don't intend to have them in there. The stored cellular products, on the other hand, could be useful and could be good depending on what they are.